Hello and welcome to this video on why viruses can survive for millennia by being frozen. While that may sound like the opening lines of a bad sci-fi horror movie, it is also true. This truth has an uncomfortable relevance with climate change causing glaciers to retreat and viruses that have been frozen for millennia to be released from their icy prisons. Most, if not all viruses, require a living host to survive. This is effectively a universal truth. The defining features of a virus include its inability to replicate outside of a host, a lack of cellular machinery, no cell membrane, and so on. These features mean a virus has no way to create more viruses without hijacking a host. Despite this rule, and the fact they have had no way to continue to create more viruses, some viruses have been found in permafrost going back as far as 30,000 years ago, and they were still able to be revived and still infectious. This makes the retreating glaciers a viable source of resurgent or re-emerging diseases. The viruses do tend to be found in one of two situations. The first is as a virus in the wild, so to speak, that is, in the soil or other core samples that aren't in a host. Because they're outside of a host, they've either had the host break down while the virus was still viable, or the virus was released into the environment. Generally, there are only small amounts in this situation, and they're not in very good condition. If they do find a susceptible host though, they can go on to proliferate. The other is when you find these viruses in the remains of a dead host. This is the slightly more difficult scenario, and it's one that we've encountered with other diseases such as anthrax. Generally, the remains are found by scavengers who then become unwitting and rather unwilling hosts. From there, the virus can proliferate. As we've seen with bird flu, SARS, Ebola, and COVID from wild animals, a disease can spread to humans, which makes this more of a concern than is apparent from the surface, or should we say the tip of the iceberg. In fact, some infections have already occurred in France. One explanation for how viruses can survive is that the viruses have different structures that are important. There are also genes which are partially responsible for how a virus survives versus those that don't. There have been studies that have tried to find out whether or not there's relationships between these, and although the structure of the Capsid envelope and the genetics aren't as important, there is some similarity with relationship to the RNA and DNA viruses, suggesting that there is some commonality in a very far back ancestor for those viruses that survive being frozen. We must also look back at why a virus is technically not alive. The lack of cellular components, and these are what would normally be a problem when freezing, don't suffer the same adverse effects when the water content crystallizes. That means there's no damage to organelles. Those same ice crystals would often also break the membrane in more complex cells. Viruses survive in part by not using organelles of their own, so these can't be damaged or destroyed, and similarly, the protein capsule of a virus is much more pliable or malleable by contrast to cells, and so it can accommodate the freezing process better, if not well. It does seem that the most important feature does seem to be whether or not the virus has a capsid. Generally speaking, envelopes are made up of lipids or fats, and these freeze at a lower temperature, but they're also not water soluble. This means that the virus envelope becomes less pliable and more prone to damage. If instead the capsid is used, they tend to be less likely to dry out, and they are less sensitive to the change in temperature. This means capsids tend to be more likely to survive being frozen for a very long time in this case, There's also the consideration of whether or not the virus has been found in a host. If it's found in a host, that body is going to provide yet another layer of protection. First and foremost, it's going to reduce the chances of the virus drying out, as most hosts are full of plenty of water. The freezing process means that they should freeze, if not quickly, slowly enough that the virus isn't going to be broken. So you're either going to have a very fast freezing process or a very slow and steady one. This means the virus won't break so easily. This, in combination with the other features of viruses, tend to contribute heavily to their ability to resist being frozen for a long time from a structural level. There also appear to be a number of genes responsible for helping viruses in cold environments, although this area is far less clear. The difficulty here is that not only does it help with infecting new hosts in cold environments, but those same genes seem to have a relationship to survivability in extreme conditions. This does sound increasingly like a game of Plague Inc., but it is true. The resistance genes responsible for harsh conditions tend to make them more survivable and infectious in those environments. There is a final concern from these viruses, and it's not related to the viruses themselves. Although they are a concern, they could also contribute to diseases in another way. That is the transmission of resistance genes. Mostly the biggest worry is antibiotic resistance, but amoebas, fungi, and others can also acquire resistances that could be a problem from genes from millennia ago that humans haven't any record of ever encountering before, and will suddenly begin becoming a huge issue for us now. Of course, not all viruses are as robust as the ones that we've seen so far. In reality, many viruses and pathogens in general will not survive millennia of being frozen. There is about a 90% attrition rate for viruses that are frozen and then defrosted. The problem, though, is one of numbers. If enough frozen pathogens are being defrosted, it will eventually lead to something that survives, and this is something of an inevitability. This is simply because so many viruses and other pathogens will be released over time that something is going to survive. When enough frozen pathogens are defrosted, and it will lead to something that survives, and it views an organism in its proximity as a susceptible host, with enough pathogens available for infection, it will start spreading. It really is just a numbers game from that point. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.